Welcome to the laboratory of Dr. Heike Daldrup-Link.
We would like to thank the California Institute of Regenerative Medicine for funding this project.
For the evaluation of new stem cell therapies, it is important to non-invasively track the injected cells in vivo. This is possible by labeling the cells in vitro with specific or multifunctional contrast agents for MR imaging or optical imaging. This video will show you how to label human mesenchymal and human embryonic stem cells for subsequent in vivo imaging. Hi, I'm Tobias Henning from the Contrast Agent Research Group at the Center for Molecular and Functional Imaging in the Department of Radiology at the University of California, San Francisco. Today I'm going to show you procedures for in vitro labeling of human mesenchymal stem cells with ferrocarbitran and of human embryonic stem cells with ferrum oxides. This technique is useful in order to localize injected cells in vivo or to obtain information about their functional state. The procedures for cell labeling with MR contrast agent varies for embryonic and mesenchymal stem cells, but both techniques involve the following steps. Preparing the cell culture, that is, plating of cells as adherent cell cultures. Preparation of labeling media, labeling of the cells by simple incubation. Trypsinizing of stem cells and washing off free contrast agent. Evaluation of labeling efficiency and cell viability. So let's get started and label some cells.